Imagine you were alive back in the 1980s and were told that computers would soon take over everything, from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. In engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it worked, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants, and animals to study and modify them for research, medicine, agriculture, and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. There are super-muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? And viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap, and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever, used for much more. The creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene-editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. Powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that can occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. The technology is certainly a bit scary, but we have a lot to gain, and genetic engineering might just be a step in the natural evolution of intelligent species in the universe. We might end disease. We could extend our life expectancy by centuries and travel to the stars. There's no need to think small when it comes to this topic. Whatever your opinion on genetic engineering, the future is approaching no matter what. What has been insane science fiction is about to become our new reality, a reality full of opportunities and challenges.